My name is Gerhard Prinsloo. I'm a lecturer at the University of South Africa in the Department of Agriculture and Animal Health. If you're looking at this video, then you're probably interested in metabolomics, and specifically NMR Metabolomics. So I'm a plant physiologist at training. I've studied medicinal plants at undergraduate level as well as at postgraduate level. After that, I've started at the ARC, where we looked at the agricultural aspects of medicinal plants, and then we realized the huge gap, knowledge gap that exists around medicinal plants, how to grow them. We realized that many of our plants are used medicinally, but we don't know which compounds are linked to that medicinal effect, and it's still a big problem currently also in our medicinal plants, that we don't know which compounds are linked to the medicine activity. And if you start looking at plants and to introduce them to commercial production, you need all this background information to answer the questions of the market. So we've introduced NMR-based metabolomics since 2012. So we had plenty of time to play around with the technique, develop it further, and also to build a bit of expertise in this field. Our research group is working on indigenous plants of South Africa, and we are utilizing metabolomics as a tool to find out what's happening in the plants. We are specifically looking at medicinal plants, also plants with cosmeceutical application, as well as indigenous vegetables. And we're looking at the medicinal properties as well as the nutritive properties of these plants. We're also looking at developing them for commercial production. So we're looking at cultivation practices such as fertilizer application, the effect of irrigation or rainfall, how to harvest and when to harvest, seasonality, all aspects like that, and how it affects the chemical profile and then subsequently the medicinal effect of your plants. So we started to use metabolomics to look at the whole profile of the material instead of just using one or two target compounds. We realized that in most of the plants that we're working off, there are only one or two or maximum 5 compounds isolated and well characterized, and that is not enough to do a proper quality assessment on this material. So we also realized when we do introduce irrigation, or if we look at different seasons, also different harvesting times, that there's a lot of changes in the plants that is not affected only, or is only seen in those target compounds. So by looking at the whole profile of your plant, you get a better idea of what is happening in the metabolism, and if there are major changes in your plant as a result of, for instance, irrigation, which obviously change your plant quite significantly because it grows faster, and there's a lot of changes on primary metabolite level as well as secondary metabolite level. So by using NMR metabolomics, we have been able to identify a lot of changes that was unexpected, that is not your mainstream results, and that's a result of looking at the whole profile and not looking at one or two compounds. There's a big drive in the agricultural field to be able to cultivate and to commercially produce our medicinal plants and our indigenous plants of the country. We have very little information on that, and by looking at different aspects such as irrigation, different soils, propagation, seasons, storage of your material, we can get a better idea of how to produce quality material for the commercial market. So NMR metabolomics is specifically a good option for me because NMR is a simple and quick method to use. The sample preparation is quite easy. It is quite simple as well. It's not affected by the environment of the laboratory, so you don't have the effect of temperatures on your samples, so you don't have the drift. It's non-destructive, so your sample is recovered 100% afterwards. And last but not least, it's quantitative, which is a very big benefit of NMR. So once you've run your samples and you've worked on your samples and you've identified compounds, you can use the exact same analysis afterwards to quantify your compounds in your sample. So that's a very big benefit. But as with the case with all equipment, there are always benefits and advantages, disadvantages of using equipment. And the disadvantages of NMR that's often mentioned is the low sensitivity of the NMR equipment, which is true. Your chromatographic techniques like LC-MS and GC-MS is much more sensitive, but there's been great improvements in the fields of NMR to improve the sensitivity. So using of a cryoprobe, or using small NMR tubes, or by using a bigger magnet or a bigger equipment can greatly enhance your sensitivity of the equipment. What we often do is to link the different techniques which give a better analysis and interpretation of your results. So by linking our NMR with LC-MS or your GC-MS results, give you a much better picture and be able to answer your biological question much better and much more accurate. The importance of metabolomics and the need for metabolomics to be more pronounced and more used in our country is so important because science is moving so fast. By not being able to determine the effect on the metabolism of plants and to provide a bigger picture and an overall holistic view of what's happening in plants, we're missing out on so much information and important aspects that we don't know about, which can only be brought to light if you use techniques like metabolomics to have a holistic view of the plants and to try to understand what's happening inside the plant, which we can't see with your normal analysis. Okay, so we are going to look at sample preparation, which is a very important aspect, especially in metabolomics. The moment you introduce human error in your samples, you see it in your analysis, and then you're never sure if it's because of the treatment or the effect that you're investigating, or if it is because of the errors that you've introduced. So what we're going to look at is how to prepare your sample consistently throughout to make sure you don't introduce human error, and to make sure that your analysis that you get at the end is reliable and your results is accurate, reflecting the biological sample and the chemical profile of your sample, and not because of factors that's been introduced. When you prepare your samples, it's important that your material and your preparation of your material is very uniform, and that starts from harvesting your material, your transport of your material, how you store it, how you dry it, what you do with it in the lab, how you grind it. You must make 100% sure that all your samples are 100% treated the same to make sure that no changes are introduced because of the handling of your material. If you've done proper sample preparation and you've get good analysis on your NMR, then there are a wider range of factors that also needs to be taken into consideration, which can be also handled in other training sessions. For instance, how to interpret your data, how to process your data, and how to make accurate interpretations, and also to answer your biological question. As mentioned earlier, sample preparation is a very important aspect, especially in metabolomics, since any changes that are introduced in the material will reflect in your analysis later on. So to make samples accurately the same, 100% the same, requires a bit of thinking and planning. And as I mentioned earlier, every step of your sample preparation is important, since from harvesting your plant material, how you transport it, how you dry it, how you grind it, to make sure that all your samples are treated 100% the same, to make sure that your changes that you see in your metabolomics analysis is just as a result of your effect that you are investigating and not because of the human error introduced. So first we are going to look at how to prepare your samples. So importantly is to weigh out your sample first. Very important is to do that accurately. We normally weigh out 50 milligrams of the material. Once again, if you weigh out more or less, you are going to introduce changes in your sample preparation or your sample extract. And as mentioned earlier, NMR is a quantitative technique. So the moment you already start with a different amount of material in your sample preparation, you will definitely be able to see that at the end in your sample because of quantitative differences. So we use a direct extraction method where you eliminate a lot of the steps with normal extraction preparations, and that is especially usable for metabolomics because it ensures that you don't introduce a lot of changes while preparing your samples. So the method we are using is developed to extract both polar as well as apolar compounds, and that is a good place to start if you don't know what is in your material, to extract your whole range of compounds from your plant. So the first extraction method that we are going to do is how to extract your apolar compounds from your material. When we do NMR metabolomics, it's important to use deuterated solvents, which means that the hydrogen atom is replaced by a deuterium. Proton NMR that is used for the analysis provides a peak for each of your hydrogen atoms in your sample. So if you have a hydrogen on your solvents, it will make a huge peak on your analysis and it will overlap your sample peaks. So it's important to use deuterated solvents. For the apolar extraction, we are going to use deuterated chloroform, and for the polar extraction, we use deuterated water or deuterium oxide, as well as deuterated methanol. Okay, so the first step is to weigh out your sample. So you're going to use a scale which is able to weigh out milligrams, not only grams. And it's very important, as I indicated earlier, you must be able to accurately weigh out 50 milligrams of your samples. We normally prepare it in a 2 milliliter Eppendorf tube because this allows for enough solvent to extract and for enough extract to go into your NMR sample. Okay, so to prepare your sample, you're going to weigh off your sample in a weighing boat. You're going to zero the equipment to make sure that you are not weighing inaccurately. So just add your material until you have accurately weighed down 50 milligrams of your plant material. Okay, so you put your sample in your 2 milliliter Eppendorf tube, and this is now ready for your samples for your solvents to be added to your samples. To add your solvents, you're going to use a pipette. When we do the apolar extraction, we are going to add 1.5 milliliter of your deuterated chloroform. Okay. So once again, it's very important to weigh out or to measure your solvents also accurately, because variation in your amount of solvent you're going to add will also influence your quantification at the end. So make sure to measure exactly 1.5 milliliter of deuterated chloroform to your sample. When you've added your solvent to your sample, you will see that there's a lot of material that is not dissolving well in your solvent. To bridge that, we are going to vortex your sample for 2 minutes. So for 2 minutes, you're going to put it on the vortex, and that is just ensuring that your material is mixed well with your solvents. So once it's properly mixed, we're going to start with the extraction process, and for this we are going to use a sonicator. A sonicator makes use of ultrasound waves, and it's breaking the cell walls, ensuring that all your compounds are extracted from your material into the solvent. So you add your samples to these floaters, you put it in a sonicator, and that will run for 15 minutes. So after you've sonicated your material, you can see that the solvent is already changed color, so your compounds have been extracted from the plant material into the solvent. And after this, we're going to put it on the centrifuge for 15 minutes at 12,000 revolutions per minute to pellet the material so that we have a clear supernatant that can go into the NMR tube. So when you load your samples on the centrifuge, it's important to load them in a balanced way so that the centrifuge is not off balance when it starts to swing. So you close the lids. You'll set your timer to 15 minutes and your speed at 12,000 RPM, and then you can start your sample. So once done, you carefully remove your sample from the centrifuge, and you'll see all the plant material is in a pellet at the bottom. And it's important not to shake this or to disturb the pellet at the bottom, because you need the clear supernatant at the top to go into your NMR tube. So what you're going to do is you're going to remove the clear supernatant from your pellet and transfer it to your NMR tube. So very carefully without disturbing your pellet, you take your clear supernatant and then you're going to add it to your NMR tube. Once you add it to your NMR tube, you can close it with a lid to prevent the solvent from evaporating, and this is now ready for your NMR analysis. So what is important is to make sure that each sample get a unique number as it's going on the NMR, and you will only receive the analysis with this unique number coming from your NMR. So we normally use a small piece of paper like this. Which you then add to your sample, and this sample name is going with your sample to the NMR.
Okay, thank you so much for that. At this point, are you guys still able to hear me?
Yes.
Thank you. So at this point, I'm going to hand over to Dr. Fadal and Prof Prinsloo, who will facilitate a Q&A session based on the sample prep video. If there are no questions, we'll move on to the next video. Feel free to post your questions in the Q&A chat, or you can then wait for the discussion session where you can then unmute and ask your questions live. So at this point, I'm going to hand over to Dr. Fadal and Prof Prinsloo. I just have another meeting to attend. I trust that you will enjoy the rest of this workshop, and I look forward to seeing everybody tomorrow. Hopefully we won't have any technical issues then. Fadal, you can take over with Prof.
Thank you, Aurelia.
Okay.
Prof Prinsloo, are you there?
Yes, I'm here. Yeah, so I think maybe just to mention, yeah, we stop now after the apolar extraction. The next video will be on polar extraction. So if there are questions on that, everyone can just hold their questions a bit, because we are going to do the polar one now after this. But if there are any other questions, you're more than welcome to raise your hand. If you can see you want to ask a question. Fidel, I don't see any questions. I think
not yet. Will you be able to share the video from your end, or I can still share the videos?
Please, if you can do that while yours is working.
Okay. Give me a few seconds then to get them. Someone asked. There is a question here, Gerhard. With the use of organic solvent with the plastic tips and EPIs not create interference?
See, we do not keep the extracts too long in that Eppendorf tubes. So I think if you're going to keep it too long there might be interference, but the time we are keeping it in that Eppendorfs and the tips is in contact for a very short time. So we also, if you have a number of samples with a chloroform, the deuterated chloroform especially, after 5 or 6 samples, you just change your tips, the pipette tips as well. Just make sure that if there is some breakdown on the plastic, that you don't get it in your samples. But It's basically for 15 minutes in the sonicator and 15 minutes in your centrifuge, and then it's removed from your Eppendorf. So it's not a long time in contact. And you also you must just make sure about the Eppendorfs you also use. There is ones that you can use that reduce the risk of this.
Another question, can the NMR be applicable in bacteria?
Yeah, sure. You see what happens is, and we'll go a bit into more detail on this tomorrow on the NMR, but your NMR is giving a peak for each hydrogen atom in your sample, and it's not destructive. So it's not, your sample is not injected into the equipment, it's staying in that tube. So anything that you add into that tube will stay there. So you can add any sample. You can use any biological sample, basically not even biological. You also use other samples as well, because it's only going to give a peak for each hydrogen atom. So you can use your bacteria as well. NMR is even used on living cells to check the metabolism of how it's progressing. So because it's not affecting your sample that you put in the equipment, you can basically analyze any sample. I see there are a few questions.
Another question, how is the pH maintained?
Yeah, so we'll do that now in the polar ones. In the apolar, we do not have really a fluctuation in the pH, so we do not adjust the pH. When we do the polar one, we do adjust the pH to 6, because that's one of the major things that might result in a chemical drift or a chemical shift. So, but we'll address that now with the polar solvents.
Okay. I think you can keep typing your questions in the chat, and also keep them for the next round of Q&A. So I'm going to play number 3. Share my screen. Can you see my screen?
Yes.
Is there a sound yet?
Yes.
Yes.
So the other method we use is to prepare the polar extraction, where we extract all the polar compounds from your extract or from the plant material. And in this case, we're going to use deuterated methanol, and we're going to prepare a buffer with deuterium oxide, or the D2O. What we add is we add potassium phosphate into the deuterium oxide, and then we're also going to add a standard at a specific concentration, which we use for quantification at a later stage. So you can take your deuterium oxide, or the D2O, so the first we're going to add is the potassium phosphate. And what is important here is to make sure that it is, the chemical formula is KH2PO4 and not K2HPO4. The one is potassium phosphate monobasic, and the other one is potassium phosphate dibasic. The dibasic is not the correct one, so make sure that you use potassium phosphate monobasic, because that will give you a pH of around 4, which we're going to adjust then to pH of 6. With sodium hydroxide. So just ensure that you use the correct buffer for your preparation. So once again, you're going to zero the scale. And in this case, we're going to add around 1.2 grams of your buffer. Which you're going to add to your deuterium oxide. Also, you're going to add a standard compound, which we call TSP, and we're going to either prepare a 0.1% TSP solution or a 0.01% solution. When you use extracts, crude extracts, it's better to use the 0.1%. Otherwise, you can just use the 0.01%. To prepare a 0.01% TSP solution, we're going to weigh off 10 milligram of your TSP, and that will be added to 100 milliliters of deuterium oxide. So this you're also going to add to your deuterium oxide, which already contain the buffer. You can shake it a bit to make sure that all your buffer and the TSP dissolved in your solvent. Afterwards, we're going to adjust the pH to 6. And this is quite important, as your pH is one of the factors that might affect your chemical shift on the NMR analysis. So it's very important to, after you've added your TSP and your buffer, to adjust your pH to 6. To adjust your pH to 6, we're going to use a 1 molar sodium hydroxide solution. Once again, it's also deuterated solvent, so the hydroxide, the hydrogen, is replaced with the deuterium. So I'm going to add dropwise your sodium hydroxide solution to your solvent to ensure that the pH is not going above 6. Okay. So once you adjusted your pH to 6, you write your details on the bottle to make sure that everyone knows that it's a buffer solution, and it's important to record the pH as well for later reference when you've done your analysis. So the other solvent we're going to use is deuterated methanol, which you do not need to prepare. Once you've prepared your buffer, you're only going to add your methanol and your deuterium oxide to your sample. So once again, you are going to weigh off your sample very accurately. You're going to weigh off 50 milligrams of your sample. Okay, and you're going to add your sample to your 2 milliliter Eppendorf tube. After that, you're going to add 700 microliters, 750 microliters deuterated methanol, as well as 750 microliters of your buffer solution. So you adjust your pipette to 750 microliters. Take 750 microliters of your buffer solution. And add 750 microliters of your methanol. After you've added your methanol and your water, you're going to vortex it for 2 minutes to mix the plant material with your solvent. After 2 minutes on the vortex, you can see the plant material and the solvent is mixed, and now it can go for 15 minutes on the sonicator. After 15 minutes, you're going to remove your sample from the sonicator. And now you can see the material is nicely mixed with your solvent. And this sample can now go onto the centrifuge. Once again, it's going for 15 minutes on the centrifuge to ensure that you get a separation from the pellet and the supernatant. Set the centrifuge on 15 minutes for 12,000 RPM. After 15 minutes, you're going to remove your sample. And you can see now you have a clear supernatant, and the plant material formed a polymer pellet at the bottom. And you're going to remove the supernatant now to your NMR tube. So once again, carefully remove your supernatant without disturbing the pellet. And transfer your extract to your NMR tube. Once again, carefully put the lid on top of the NMR tube This prevents the solvent from evaporating. Important is also to just label your sample. Which you do by just writing the sample name on a piece of paper. Pushing the tube through the piece of paper, and this sample name will go with your sample on the NMR, and your analysis will be done with this sample name.
Thank you. I will play the next video now. Can you see my screen?
Yes, we can.
Yes, we can.
The sound?
Yes.
Yes.
Okay, thank you.
Okay, so this is a 600 megahertz NMR equipment. The samples is going there in the top. It's pulled down into your equipment, and in this casing here is a lot of magnets, which creates this magnetic field around your sample. And we will later on explain how your magnetic field is giving you peaks on each of your hydrogen atoms. Okay, so once you have your NMR samples ready, we can now put it on the NMR. So the first thing that we need to do is that we need to put it in the holder. So you push it through like this. And this is just showing you the area where your sample will be measured. So when you add your sample to your tube, just make sure that that whole area is filled with your sample. And this is then ready to go onto the NMR. So this sample is now going into the auto sampler, and there it's ready for analysis. So once your sample is inside the NMR, it will be analyzed and your data will be sent to your computer. The settings on your computer is very important for the equipment. So one of the things that you have to make sure that is correctly set up is your shimming, as well as the tuning of the equipment, to make sure also that your samples are consistently run on the same settings. When you run your polar samples, when you use water as your solvent, you can also use the preset setting, which will take out the water peak, especially when it overlaps some of your some of your sample peaks. For the chloroform, you don't have that because there's no water in your sample, so there you just do a normal run. What is important also is to check your sample when you have your analysis on the computer. For the resolution, you should have very good resolution on your samples, as well as your reference standard should be at 0.0. For checking your resolution, one easy way is to do that is to check your methanol peak. When you have bad resolution, it will just give you one broad single peak, whereas it should be 5 separate peaks. And that is a clear indication of how good your resolution is. If you have good resolution, then you can continue with running the rest of your samples.
Thank you. I will hand over to Prof for questions or anything that you need to add on the videos.
Yeah, I think we can open up for questions on the sample preparation. I will also quickly just go back to the chat to see if there are questions there that we haven't answered. Okay, so there was a question on the solvent. So yes, so deuterated chloroform for your apolar extraction, and for the polar extraction we use methanol water, all deuterated solvents. And those samples we normally, those solvents we normally store in the fridge, so it's not necessary to take special precautions there. Yeah, so you can use other organic solvents depending on your samples. So there are deuterated acetone. I think there's also deuterated ethanol. But what we've seen is that it's best to stick to one method. If you do very precise and very focused on one or two compounds, maybe then I think switch to different solvents. But it's best to stick to one method. We've seen with some of your projects, a year or two later, you run samples again, and then you want to combine, or you want to check, or you want to compare with some of your previous samples. And if you've then changed your method, then you can't do that. So, so also when you have a group of people that work together, one or two students or staff members, if you standardize your method, then everyone knows how to use it and what is used. And then it's comparable between your samples. So you never know later on if you want to compare it with other samples or a different data set. So, so my advice is to just stick to one method. Develop a method. This one has been developed by the University of Leiden in the Netherlands, which works well. But I would not advise to switch to different solvents very often. Okay, why do we adjust to a pH of 6? It's not specific to pH of 6. You can also adjust to a pH of 7. As I mentioned earlier in the videos, the pH is one of the things that can result in a chemical shift change. So once again, stick to one pH and keep to that. Just do not switch or change your pH of your solvents between your projects or between different people of the group, because that will result in not being able to compare results afterwards. 6 is a general pH to use. So as I say, this method has been developed for a while now, so if you stick to a pH of 6, it's fine, but it's not cast in stone. Okay, the internal standards, you will see when you buy deuterated chloroform. I don't know, I think most of them have already the standard in there, TMS. So you don't need to add an additional standard there. So with chloroform, deuterated chloroform, you already have your standard at 1% added to your solvent already. If it's not there, then you have to additionally add that at 1%, but normally just check for the solvent that already has it as part of your solvent. Yeah, so you then use that team S at 1%. You will then use for quantification on your polar solvents. Okay, then this question from Aurelia. A dual polar and non-polar extraction at once. The method was developed to keep it separate because I think it's easier when you separate the different polarity compounds from each other so that they don't overlap all at the same time. But I think it would be possible to do just one extraction, but I would guess that you will have a lot more overlap of your peaks, especially in the region 0 to 2, where a lot of your single your carbon chains are absorbing in that region, which might overlap with some of your, for instance, your amino acids that also absorb in that. So yeah, it will be possible. I don't think it would not be possible, but it will depend on what you on your purpose or what you are looking for. Any other questions? Okay, I see there's another question. Since methanol can extract some of the compounds chloroform will extract, why not use methanol? Yeah, I just think it's chloroform is targeting the more apolar compounds on that polar range, where your methanol water is targeting the more polar compounds. So yes, your methanol will be able to extract some of your compounds that will go into chloroform as well. But chloroform is definitely going to extract more of your apolar compounds. So I think that's why chloroform is just used. What you try to do here is to extract your whole range of compounds. If you don't know what you are working with, once you start with a sample and you do your metabolomics, you're going to compare your profiles after different treatments or the effect you want to investigate, and you don't know which compounds will be affected. So what we try here is to extract as much as possible from your plant material, and these two methods, the polar and the apolar, is quite simple and easy. So the trials that we've done is to check a range of solvents and a range of extraction methods, and this method was the best method to extract a whole range of compounds from apolar to polar, and to keep the steps and the number of samples to a minimum. So yes, you will be able to extract with methanol, but this method is specifically targeting the whole range of polarity compounds.
Okay.
Welcome back, everyone. So this morning we've done sample preparation, a bit of basics and background on metabolomics, NMR Metabolomics. So the next one we're going to look at is experimental layout for NMR Metabolomics. And Dr. Lufuna Nemadotsi is going to handle this. She is also a lecturer at UNISA in the Department of Agriculture and Animal Health. And She did a PhD on a project on metabolomics, and since then has been involved quite actively with the research group as well. So, so I'm going to hand over to Dr. Nemadotsi. Funo, are you here?
Yes, I am here, Prof.
Okay. Do you want to share something?
Yes.
Okay. Yes, presentation is loaded. You can continue.
Good morning, everybody. I am Dr. L. Nemadotsi. You can just simply refer to me as Dr. L. And I'm going to be talking to you about experimental layout for NMR metabolomics. I'm just going to give you a bit of a background on the metabolomics. Sorry about that. So there's been development of diverse range of independent instrumental techniques which can be used in metabolomics, techniques such as liquid chromatography, gas chromatography, mass spectrometer, and your high performance liquid chromatography. But I'm going to focus more on the NMR, which is the nuclear magnetic resonance. NMR just started getting recognition in the year 2000, and this was due to the number of lot of publications that were published in the field of metabolomics using NMR. So more scientists started, getting interested in the NMR, and that's where it grew. But of course, as Professor Prinsloo has alluded, at UNISA, we started using NMR in the year 2012. Why NMR? If I have to ask, if maybe you're asking yourself why NMR, because it will give you the highest quantification of compounds. It just in a simple analysis, so you can get a lot of quantification using NMR. Okay, the other reason why we're using NMR is because it is fast and is highly reproducible. I'll just give you an example of what do I mean by highly reproducible. If you have run NMR samples or analysis at CSIR, which is where we usually do our NMR, anybody who's anywhere else, whether it's UJ or overseas, must have the same results that you got. That's why we're saying it's highly reproducible. And also it also helps in identification of the major metabolites in a single analysis. And we use NMR because we are trying to investigate the differences between the metabolic or the metabolites that are found in a sample, can be a leaf sample or a cell sample or anything that is plant related. So we use NMR to investigate if there is a differences in between the sample. And we also use NMR we also use NMR in the metabolic profiling, which is also known as metabotyping, so that we can understand the responsible metabolites that or the responsible metabolites in the pathway of that give the pathway of the metabolites that are found in that particular samples or compound. So in a nutshell, NMR will give you a wealth or a lot of information on the complex or information of the complex of metabolites that are found in a particular sample. So you end up having a lot of, rather, I'll say in a simple language, I'll say you will be spoiled for choices when it comes to the complex of compounds that you're going to find. But ideally, an NMR spectra will have something like 64,000 peaks that are going to be found either in a complex biological fluid or a cell of tissues. Where do we start when we are doing this NMR? Where do we start? Of course, I'm not going to go much on what Professor Prinsloo has said, but on NMR, we need to, first and foremost, you need to design your experiment and the sample strategy. So this way you're going to determine how many samples are you going to run or to analyze. And remember, we always say in metabolomics, the more samples, the better. So from 50, I would say 50 or 60 samples, you can actually have something good to report about. And then Professor Prinsloo also took you through to sample preparation and extraction. I was very happy that, there was a video, so you guys now understand what sample preparation and extraction is all about. Remember, you need to be consistent with your the solvents that you are using. Things like your pH, I'm going to talk more about that. Things like your pH, your buffer, and all of that. Everything must be consistent. You cannot alternate. You can't be using chloroform and methanol next, chloroform, and no. If you choose to use one solvent, stick to that and continue until the end. If you find that the results are not as good as you wanted, then you can now change maybe to use another solvent, alternate perhaps, or use another. And then so after we've done that, what we're going to get raw data. That is the data that we're going to get after the, I think the last video that Professor Prinsloo showed was after he had put the supernatant into the NMR. So what's going to happen there, we are going to get raw data. So now this raw data, we need to process this. We do this using Mastronova, where we have to process, it involves things like your normalization, making sure that the peaks are the peaks are positioned well, the referencing, you phase correct and baseline correction, all of that. And then once that is done, then we need to analyze the data. So we analyze the data. We have our own statistical analysis that we use, which is called a multivariate statistical analysis tool. Somebody might be saying, why do we have to use a multivariate, and why can't we just use ANOVA as an example, or one way or two ways ANOVA? No, we don't use that because we use multivariate because it gives us the proper biological interpretation of the data that we found, the metabolic data that we found. So for us to actually have to come to a point where we can say we have data, we need to analyze it using, we've got 2 or 3 models, statistical model that we use. So the first one is PCA, which is the principal component analysis, and then we also have the OPLSDA or PLS. I'll tell you the difference between the two. So the PCA is the unsupervised model. So in other words, you're going to take the data that you have found, that you have after you're pre-processed, as is. You do not extract or reduce anything. You just take it as it is, and then you run it. Then it will give you, if it can show you the either a good separation or a distinct separation of the variation of your compounds, of the metabolites that you found in your sample. That is PSA. Now, in the event where you do not find anything that you are looking for, you can now go to OPLSDA. OPLSDA is just a supervised model. This is where now you can either remove maybe some outliers. I'm going to show you on the next slide, but you're going to remove some of the outliers, some of the unwanted noise, what you feel like you don't need, you remove it, and then you run it. That's when you're going to get a good separation or a distinct separation of the compounds. Once that we have that, I'm sorry about that. So once we have that, then we are going to now have the information, what I call, either we are interpreting our data. We need now to interpret the data. Okay, so the data that we found, to a lay person, when you are now explaining, he or she must be able to understand, oh, so this is what you found, or this is what your data means. That's about interpretation of data. And with that, then we will have enough information to say, do we have targeted or targeted data. Professor Prinsloo touched a little bit on the targeted and the untargeted. So if you don't know what you're looking for, those are, we call them untargeted metabolites. If you know exactly what you are looking for, then we call this your targeted. I hope I'm not confused. Yeah. Okay. So on the next slide. Okay. So this is just the previous. I took this from my as Professor Prinsloo said, I took this from my PhD study, and I was working with soil. So this is just the extraction method that I used. I collected the soil, and then we used methanol, and we did all the processes, the sonicating, the vortexing, and then all the way to transferring the supernatant to the NMR tubes, and we closed them, and then it was taken to NMR, and then the Mastronova that I was telling you that we used for pre-processing our data. So this is just to show you guys what I was talking about. I don't know what's going on. Okay, so this is, remember I said we have two statistical model or three statistical model that we use to analyze our data. So in this one here, we had started with PCA, but it didn't give us exactly what we didn't give us. There was really no difference. So we had to now use move to OP-LSDA, and we used OP-LSDA where we had to supervise to like remove some of the things that were making noise, or what we call noise. And as you can see, there's a good separation. This is, if I have to just give you a brief of what this means. So what happened is I worked with this tree called Bouquet africana, which can only grow, or which is known to grow in its natural habitat. So farmers had tried for so many years to grow it outside its natural habitat. And what happened would be it will grow for a period of 6 to 8 months, and then it dies. So when I came in, we brought in NMR, and with the help of NMR, we were able to see, or rather to find the difference between the two soils. When I say two soil, I mean soil where Bouquet africana grows compared to where it does not grow. So we took the samples from where it grows to where it doesn't grow, and then we ran NMR separately, and it showed us this, what is showing in this screen now, where the one below the x-axis is showing the compounds that were found where Bouquet africana trees does not grow at all. And then the one that is above the x-axis shows the compounds, or these are the metabolites that were found where this tree grows. So as you can see, NMR has a way of unearthing it has a way of bringing solution in things that were that has been a problem and that didn't have solutions for the longest time. So for us, that was a groundbreaking study. Okay, so this is then okay. So after you've gotten the spectra of NMR, then what? What do you do? This is just all these answers. Rather, it talks back to what I was showing you about the biomarkers at the peaks. Professor Prinsloo also showed you a peak of the 5 peaks and whatnot. So where you have to check if the resolution is right. The peaks, are the peaks really, are they biomarkers or are they noise? You check those things, but you learn with time. If the peak is, if the reference or the standard is not has not started on 0, then you need to clean your data. This is about cleaning your data, and then after you've seen the peaks of your found the right peaks, you also need to check the signal intensity or the position of the peak. How high is the peak? Where is it positioned? And then you can now say, with this peak, I think there is something. If you try to go through it and you use things such as your economics to either confirm your findings, then you'll be able to now interpret or find or make sure or be certain that there is a pathway and then there is a significant separation. So it's a process that follows one after another. You start by pre-processing. After pre-processing, you do a statistical analysis. After that, then you now check if there is a significant differences or isolation or separation. From there on, you move to the next step, which is now the confirmation of what you have found, what you think is there. And then with the use of genomics, then we need to now either identify or isolate. So if you need to confirm the compounds, not just by saying this is it. No, you confirm it with genomics, and then you can confirm it also, further confirm it with human metabolic database. The good things about NMR is that it is robust and it's highly reproducible. I've told you that we had to send my data to Wageningen University where it was run, and we found the same thing. So if you run NMR at UNISA and somebody's running it at University of Venda or UJ and they don't find something, they don't find the same thing, you have not done something. Something was not done right. Something was not done. So it is reproducible, although we know that it's also less in fact, I'm going to talk about that on the next slide. So the other one is that it will give you specific structural information about the metabolites that are there. It is not going to, it tells you exactly what it is. The number 4 is that it only requires minimal sample preparation. So you're not going to spend a lot of time by preparing your samples, of course, because you saw what Professor Prinsloo was showing. It's not much, but of course, with the number of samples that you have, it might take longer. So, so the more samples you have, of course, you must just be ready to be standing in the lab and be doing the sample preparation. And of course, like I said initially, that it is highly quantitative, which means that you are going to get a lot of metabolites, some that you think may not be important. So you can just leave them out and only focus on the ones that you think are important or are distinct. I think I also need to indicate that NMR has been used in a lot of studies, such as it will give you quantification metabolites such as your amino acids, your organic acids, phenolic compounds, your sugars, and lots of different metabolites that you can get using NMR. The bad thing about NMR, I call it bad because once you make a mistake, Professor Prinsloo spoke about it, and I saw somebody also asking a question, why 6? So this is the pH adjustments. Once you make a mistake with the pH adjustment, it actually precludes the shift, or the shift, the chemical shift and the chemical composition. So once the pH is wrong, if you put it on 5, it's not going to give you a good chemical shift. You need to make sure that your pH is at 6 or is at 7, as Professor Prinsloo said. And once it's at 6, do not shift it again, or rather don't alternate it to 7 or 5. Make sure that it's at 6. And then I cannot talk about the bed and not come with solutions. So the solution is that you need to adjust it before you do anything, and make sure that your pH is adjusted. I also read this other article where they speak of specific solvents that come with the right pH. So if you are using methanol, you look for methanol, or if you are using methanol and the buffer, it should be, there are those solvents that comes with the specific pH that you need. In this case, we said it's 6 or 7. Conclusion. So some of us, we did NMR when it was just, especially in South Africa, because it has been used in developed countries for the longest, since I said 2000, since the year 2000, and here we only started it in 2012. So you can just imagine, as you know, in research, we don't stay in one place. We revolve and we evolve. New ideas are coming. So there's more advanced NMR spectroscopy methods that are now developed, or that has been developed. There's also more analysis software, For example, we use 600 megahertz at CSIR. There are 1,000 something megahertz, and so the bigger the software, it means that you can run more samples. And we are also praying, Professor Prinsloo is here, is listening, that we get a bigger soft NMR, because we are also getting many students who will be doing NMR. So I think the bigger NMR will do. And also bioinformatics, informatics tools are also advanced, advancing. And even with the pre-processing data, there is more advanced pre-processing softwares that you can use instead of just your menstrual over. So what you need to do is just to equip yourself, read as much as you can, find out what is happening in other countries, like in other developed countries, what are they using. And this means that it also opens more ideas for collaboration with other researchers who are in developed countries who have been using NMR for the longest time. So I'm sure we can learn from them. The other thing is the analysis strategies are also involved within the field of within the field of NMR metabolomics. So in as much as we say that NMR is less sensitive than LC and GCMS and the high performance liquid chromatography, I believe that in no time this will be at par, because most researchers are now excited with NMR and they're coming up with new ideas. And we're hoping that you guys are also going to be coming with new ideas by the time you finish your PhDs and your masters. I would like to acknowledge Professor Prinsloo for organizing this workshop, and yeah, for allowing some of us to also, form part of Metabolomics Society in South Africa. And I also like to acknowledge Professor Verwoerd. Even though he's late, he really worked hard to make sure that UNISA is where it is when it comes to metabolomics studies and research. And yeah, he has really played a huge role, of course, with the help of Professor Prinsloo. So I would like to thank those two. And I also like to thank you guys for attending. I see we have something like 102 people attending. So this is the first, and I'm grateful. Thank you.
So thanks, Lufuno. We can open up for questions then. Maybe let's just start on, while Lufuno is here, maybe questions on her presentation. I will go to the chat now and have a look at the questions there. But maybe if there are specific questions for Lufuno. Okay, while waiting, okay, there's a question I see here on the use of solvent. Is it not better to optimize it rather than one for all different samples? Yes, I think the method that we shared with you is what you do when you start with this, and it's quite a general method, so it works well. So if you then find something interesting or so, then you can refine your method and optimize it. But I think at that stage, you will probably involve other methods such as LC or GC or some of your analytical techniques. So the methods that we used here is really well developed, and it will give you really good results. I can almost guarantee you that you will get good results on these solvents. And once you find something interesting, then yes, then you can isolate or focus on one or two of your compounds or your biomarkers. But that's down the line. To first get your NMR analysis, your NMR metabolomic analysis, these two methods will get you quite a good results already. Okay, the Mestranova. Basically, there are 4 steps that we normally do. It's to make sure that your reference is on 0, that you normalize. We put the NMR, the standard, to a specific level in all your samples. So you kind of standardize your standards. You make sure the baseline is correct and so on. But that we'll do on Thursday. On Thursday, when we start, we will start looking at how you pre-process your data. We'll give you some data that you can work on yourself. So as I mentioned, if you can download Mestranova in the meantime before Thursday, then we're going to show you hands-on on screen what steps you take and how you pre-process your data, and how you develop your database that you're going to analyze on Simca or your multivariate net analysis. So on Thursday, we will take you through all these steps, one step at a time. Make sure that you know how to pre-process your data when you leave this workshop. Yes, I will post the software. We call it Mestranova, and the other one is Simca. So it's those 2 softwares. For both of them, you can download the evaluation version. If you don't have access to it, if you don't have it yet, you can download a evaluation version. I think it's active for 3 months or something. So, but I think for Simca you need to get a code or something. So just don't do it Thursday morning, then you might not have it in time. So maybe this afternoon or tomorrow morning, just please send, yeah, just try to download. Yes, thanks, Wilson. That's Mestranova, and the other one is Simca. Wilson, if you just also write there Simca as well. So those are the 2 softwares that you need to download. So if you just go to Google, you will find the softwares Mestranova and Simca. There's it. Thank you. Okay. Any other questions? If you want to ask questions, just raise your hands. Sometimes it's easier to just ask the question and not type it. And it might be something that the others are also concerned about or not sure about. So if you have any questions, let's just post them. But if not, Fidel, I think we are basically then done. There was also mentioned that the videos will become available, so if you want to watch them later on again, at least you don't have to watch yourself doing the videos. So it's quite terrifying when you have to watch yourself on videos. So, so I'm not going to watch them again, but you're more than welcome.
Any other question or comment or needs that a point that you would love or like for them to touch on either tomorrow or Thursday?
Prof, I think I'm gonna just ask some few people. Shadrach and Abel, don't you have a question? Or Daphne?
No, I don't have a question on my side so far.
I'm also sorted, Doctor.
Yeah. Okay. Yeah, I think it's fine. I think tomorrow we'll go a bit deeper into the basics of NMR, so that you can get an idea of when you get your analysis, what you will see. And then also we'll have a look at the interface of NMR and other equipment such as the LC and the application of NMR. And then we'll give you nice examples tomorrow afternoon. Lufuno touched on one of the examples that she worked on. Maybe she can give a bit more details on that yesterday, because that was on soil. So that's a different application. Earlier there was a question on bacteria instead of plants. So so you will see the application on soil was very effective. So you can do it on any samples basically. So we'll give some examples, and then on Thursday we'll go through all the steps: how you pre-process, how you get your database, how you put it in Simca, and then your interpretation of your data. So we'll go through the whole process in the next 2 days. I see there are questions now. Okay, so databases, Lufuna also touched on that. There's no real database for plant metabolites, so we make use of the human metabolome database mostly. That's where we start, and then we make use of things like Genomics, which is also based on the human metabolome database. But there's a lot of overlap between your plant metabolites and your animal metabolites. And that's a good start. So we start with that, see for any matches, check for any matches on the databases. And then from there onwards, we go deeper. And that's where this previous question on different solvents and so on comes in. Then you can optimize. So when you find your biomarkers or compounds of interest, then you will start to zoom in on those ones and then develop your method further. So the database, so we basically use, you can check that, human metabolome database. It's also very applicable for your LC-MS, but they often, especially for your major compounds, which is a nice feature, we can show that's already also for you on Friday, on Thursday. It shows you the NMR profile of pure compounds. So that's a nice feature to be able to see what the profile looks like and how you can match it to your profile on your analysis. We'll give you a few tips on how to make sure that you're not overestimating or giving incorrect annotation of your compounds. So if you can also check that in the meantime, the human metabolome database. If you go to a compound, put a compound in there, and then you'll see there are, most of them has got LC-MS data, but many of them also NMR. So that's quite nice to use. Okay, and there's then the link to the YouTube page where the videos will be loaded.
Yes, Dr. L.
Yes, I wanted to say that it's just in addition of what Professor Prinsloo was saying, that there is no database as yet, and in South Africa there's actually nothing. So I think should this grow and we get more students, either UJ or UNISA, get more students who are working on metabolomics, we would be able to have our own library, library data, and because I do not know of anyone who's working on human metabolomics in South Africa as yet. So should we continue with the plants and the soil, we might actually be in a good position to have our own metabolomics database in South Africa.
Yeah, well, the database always, I mean, that's a dream of everyone to have a nice database, especially for plants. But for now, we use what we have. But yeah, I mean, once we can grow metabolomics, and especially the NMR Metabolomics, the guys at Northwest is doing a lot of human, but also mostly LC-MS based, a little bit of NMR. But yeah, the NMR databases, I think, are a bit lacking. So yeah. Okay, I didn't see any other questions. Fidel, I think we are.
Yes, if there is no more questions, thank you so much, the team from UNISA, Prof Prinsloo and the group. Thank you all for attending. I think tomorrow and the Thursday, there will be more information, and I think that also we bring up more questions in terms of how to conduct successfully NMR metabolomics. The links, I think they have been shared for tomorrow and Thursday sessions. Please follow up. Aurelia will coordinate tomorrow's sessions. And I think, Aurelia, you are typing something. I'll give her a few seconds and see. Okay, yes, please sign in tomorrow at 9. The session starts at 9 a.m. Thank you all for attending, and have a wonderful afternoon.
Yeah, maybe just as a last, sorry, chipping in again. Yeah, we'll start with the NMR basics tomorrow morning, 9 o'clock. So please don't be late, otherwise you're going to miss the first part, and that is kind of building on top of each other. So so please try to be on time tomorrow morning, 9 o'clock, that you don't miss the first part, because otherwise you will miss the whole basics, the foundation of what you're going to see later on in the NMR metabolomics. Okay.
Thank you so much.
Thank you. Bye.
Bye.
Good. Thank you. See everyone tomorrow.
Bye.
Okay, bye.
Bye.